So here we're going to run through the neuroanatomy of sleep and wakefulness. I particularly enjoy this topic because I'm sleep medicine fellowship trained in addition to neurology, so I like the underpinnings here. We're going to talk about the biology of sleep and wakefulness, and then non-REM and REM sleep physiology. The different biological topics here are suprachiasmatic circuitry, orexin and the flip-flop switch, and then wakefulness neurobiology. So let's start with suprachiasmatic circuitry with melatonin release. Melatonin release induces sleep. It's produced and released by the pineal gland here. So the circuitry or the neuronal pools we're interested in come from the paraventricular nucleus and suprachiasmatic nucleus in the hypothalamus, the superior portion of the spinal cord here, and the superior cervical ganglion. Now we're going to talk about a dark phase and a light phase. In the dark phase, we're induced to sleep. In the light phase, we're induced to not sleep, to be awake. So the paraventricular nucleus has projections to the spinal cord and then via the superior cervical ganglion to the pineal gland for production and release of melatonin. This is all part of the dark phase. We inhibit the dark phase via the light phase. So when light is captured by the retina via the retinal hypothalamic pathway, there's innervation of the suprachiasmatic nucleus, which inhibits the paraventricular nucleus. Think of the suprachiasmatic nucleus as the optic chiasm. So light is received by the optic nerves. Suprachiasmatic nucleus sits near the optic chiasm, so it makes sense that it would be part of the light phase. So now let's talk about orexin and the flip-flop switch. This is a stabilizer of states. So sleep and wakefulness are two separate states, like the lights being on or off. They're binary. We want the person to be stabilized in one state or the other. When they're unable to be stabilized in either state, we call this narcolepsy. So orexin is really key for this. If we want to move from sleep to wakefulness and maintain wakefulness, the orexin area stimulates wake-promoting cells and also inhibits the sleep center. In contrast, if we want to remain in the sleep phase, then the sleep center inhibits these wake-promoting cells. So what are the wake-promoting cells? Well, there's a variety of different cells in the frontal lobe here, cholinergic cells in the basal forebrain here. In the hypothalamus, out of the tuberomammillary nucleus, we have histaminergic cells. And then within the brainstem at different levels here, in the midbrain, we have dopaminergic cells, as well as cholinergic cells. Out of the pons, we have noradrenergic, adrenaline, and then along the vertical axis of the brainstem, we have serotonergic cells. So these are all different neurotransmitters that help promote wakefulness. If we look for a pharmacologic correlation, however, it is a little bit trickier than at first blush. Cholinesterase inhibitors maintain acetylcholine in the system, and they promote wakefulness. So there's a nice one-to-one there. Tricyclic antidepressants inhibit histamine and inhibit acetylcholine, so they produce sleep. So we give tricyclics such as amitriptyline or nortriptyline at bedtime to help promote sleep. In addition, amphetamines promote wakefulness, so we give them during the daytime or in the morning. But things get a little trickier with, for instance, dopamine. Dopamine agonists actually can be soporific, can be sleep-inducing, despite the fact that dopamine itself is generally considered a wakefulness neurotransmitter. As well, even though serotonin is considered a wakefulness neurotransmitter, SSRIs, things that increase the amount of serotonin, make us sleepy. So there's clearly other layers when it comes to receptors, various subtypes of these neurotransmitter receptors as to how different drugs act on these wakefulness neurotransmitters. So let's move to sleep physiology. There's a great story in terms of how von Economo was able to take patients with encephalitis lethargica, patients who had sleep disorders following the influenza. He was able to look at cadaveric sections and correlate clinical manifestations, whether they had hypersomnia, oversleep drive, or insomnia, the inability to sleep, and then see what was going on in terms of areas of injury in their brain. And what he deduced was that the anterior portion of the hypothalamus produces sleep, and then posterior and into the brainstem, which we saw, tuberomammillary nucleus and into the brainstem, produces wakefulness. So now let's look at the physiology of non-REM sleep circuitry. So the thalamus is the core generator we're going to focus on. The reticular nucleus via GABAergic neurons inhibits these thalamocortical neurons. When they reach negative 65 millivolts, T-type calcium channels open. This is a high-yield board topic, producing this low threshold spike, and then there's refraction. So via this setup, this physiological setup, we can see EEG changes via activation of the cerebral cortex, these various sleep patterns on EEG. There is further excitation of the reticular nuclei with this inhibition here. And then there is inhibition of excitation of various thalamic neurons. So for instance, the lateral geniculate nucleus is part of the visual system. When we're asleep, we don't want to be using our visual system. We don't want to be aware of various things in the visual world because that's going to wake us up. So we gate out this information. This is called sensory gating. Let's move on to REM circuitry here. So this is fairly complicated circuitry. We have a REM promoting region in the pons. We have an inhibition center in the midbrain, and then we have disinhibition centers coming out of the hypothalamus and the medulla. And then we have these specific neuronal pools, which you can learn on your own. There's inhibition via the hypothalamus and medulla on the midbrain, which is inhibiting the pons. So there's overall disinhibition here. And the excitation is both to the cerebral cortex, and we can see specific patterns on EEG, REM patterns on EEG. And then there's excitation of the supraolivary medulla, which causes muscle atonia. So during REM sleep, during dream sleep, we're effectively paralyzed, we're unable to move, and this helps prevent us from acting out our dreams. And there are REM sleep behavior disorders where patients do not have this muscle atonia and they do act out their dreams. So that's a quick trip through the neurocircuitry of sleep and wakefulness. A lot of really nuanced information there that is quite fascinating to learn.